Okay.
All right. Hi, everybody. It's my pleasure to introduce Surya Gayet. Surya obtained his PhD in experimental psychology from Utrecht University under supervision of Stefan van der Stichel. And I think I'm probably the only person in the audience who could pronounce that. During his PhD, he also did a research visit in Berlin to work together with John Dylan Haynes. And after his PhD, Surya went on to pursue postdoctoral research first at Utrecht University and then at the Danvers Institute, where he worked together with Marius Pele, and that's also where I first met him. Now, since 2021, Surya has been tenured at Utrecht University, where he leads the Consciousness, Attention and Perception, or CAP lab, which is also a pun, as you can see, and there's also a logo. And in his research, Surya combines rigorous psychophysics with state-of-the-art neuroimaging, and he combines all of this also with a very strong emphasis on bringing science to the public through various outreach activities, which I always think is very cool. And today, Surya will talk to us about tracking perceptual competition with rapid invisible frequency tagging. So, Surya, please take it away.
Thanks a lot, Nadine, for the very, very kind introduction. So I was thinking for a bit what I would want to present here, but we have been starting this sort of very new and exciting line of research here as a sort of a sub-lab within my CAP lab, which uses rapid invisible frequency tagging, a relatively new technique to track perceptual competition or visual competition. And there's now like 3 PhD students working mostly full-time doing rift experiments. It's a promising technique, and today I hope to present like a couple of exciting projects that I think can give us some understanding that maybe other techniques cannot. So to start out a bit, of course, we're all familiar with the idea that whenever we're awake and our eyes are open, there's so much information that we need to somehow make a selection out of it. And unlike a camera that sort of devotes an equal amount of processing resources to every single pixel on the screen, we do this very differently, right? Instead, our visual system selects part of the visual world to favor for visual processing. And one way of doing that is just to foveate that location, right? So we make an eye movement to that location, and then we can sample from that location more optimally. And it's very nice that we can do that, but it's also quite effortful. So for instance, if you try someday making either horizontal or diagonal saccades, moving this direction or this direction, you will immediately feel that one is more difficult than the other, showing that there's actually some substantial costs involved, metabolically speaking, for something that you're doing quite a lot. So something that may be cheaper than that and maybe more rapid, but we can discuss this, of course, is to use covert attention. So to basically keep your eyes at the location, but favor one part of the visual field at the expense of the other internally. So basically using your software rather than your hardware, if you will. So covert attention, sort of this means of biasing one bit of the visual input over the other, has been a huge topic of investigation, as you all know, of course. But there's also a problem with studying covert attention. Typically, this is done by, for instance, comparing like an unattended or less attended location with a more attended location, and then you expect better performance or perceptual discrimination or decodability or whatever at that attended location. But if you present something at the attended location or at the unattended location, you also change the normal allocation of attention, right? Even if participants know that something may appear at some location that may be probed, this is no longer an attended location. So measuring it inherently disrupts the normal process of perceptual or attentional competition. So then the question that we had is how can we track something as quick and as elusive as covert attention while not interfering with it, while not presenting any visible stimuli. And this line of research, which bases its method on rapid invisible frequency tagging, I will briefly introduce to you what it is. So maybe you can think along with me at the end what we're actually measuring with it. So it's based on a very simple concept that whenever there is first no stimulation and then there is an event and then stimulation arises, right? This event could be auditory, it could be visual, it could be whatever you want. Then if you measure some electrophysiological or some other response, then you will have a response to this event. And this is the basic idea of ERPs. Now, what happens if you, instead of having one event, you have a continuous changing event, like a burst of events, then this is what is known as SSVPs. Now, the following slide may contain flashing images. I'm not sure if that's also the case on Zoom, but let's see how that works. So the idea there is that you flash an image, so it should be flashing this black circle, and then because of that, whatever brain signal you recover when the temporal resolution is enough should also have this exact same frequency in it. So if you flash a circle at 15 hertz, you should find a clear peak at 15 hertz. And then the fun thing is if you flash two different stimuli at two different frequencies, you will recover both from the signal. And then in this case, you have Mario, you have Luigi. If I tell you to covertly attend Luigi, because Luigi is relevant for some subsequent task, then you'll see that the amplitude of the 15 hertz response, which is specific to Luigi, will be larger than the amplitude to the Mario frequency. Okay, so this is the idea of SSVPs, which stands for steady state visually evoked potentials. Now, the typical flicker tasks or flicker frequencies that are being used in these SSVP studies are around here, around 10, 20, maybe 30 hertz, like alpha and beta range. But the issue there is that flickering is visible. And if I'm flickering a couple of things at 10 hertz, 20 hertz, 30 hertz in your periphery, this is quite disruptive to sort of your normal allocation of attention. Now, on the other hand, if I would flicker something at like 100 hertz, there's barely any signal to recover in the EEG, in the MEG, or other types of fast recording methods. So in between that space, we have sort of this Goldilocks area, which is where we use Rift, where we are actually able to retrieve a signal from the EEG or MEG, but people cannot perceive the flicker. And to achieve that, I think there's like 5 or 6 labs in the world that are currently doing this. Most people use this VPIX projector, which goes up to 1440 hertz and allows us to get nice sinusoidal modulations of luminance of multiple frequencies across the screen. And 1440 hertz is a bit faster than your normal monitor, but this is really what you need to be able to tag multiple locations at the same time. Hence the rapid invisible frequency tagging. Now, in this situation, you would have a Mario and a Luigi that are tagged, but imperceptibly. People just see two stimuli on the screen, but still, if you would attend Luigi over Mario, you would find a stronger, well, let's say, Rift response now to Luigi as compared to Mario. And this makes it sort of an invisible marker of perceptual competition or an invisible marker of covert attention, if you will. Now, as a side note, instead of like tagging stimuli on the screen, which is also very useful, you can also tag gray background by sinusoidally modulating the luminance between black and white so that you end up, if you do your luminance calibration properly, with just a bit gray background area, but which is actually being tagged at 60 hertz, 64 hertz, and so on. And then you can recover later the responses to these different locations on the screen, although the participants just see a bunch of gray areas. So that is the general idea behind the method. But of course, we don't do it just for fun. We also do it to study perceptual competition and to answer some questions that we're interested in. And today I'm going to briefly describe 4 projects from my lab, and if there's some time, maybe share some future perspectives without eating too much into the discussion time afterwards. And the first one that I want to go to is about competition between top-down goals on the one hand and bottom-up saliency on the other hand. This is a project that has been led by my PhD student, Dan. Now imagine you're driving your car, you're looking at the road ahead, but then something might pop up that is quite salient, like a billboard or something else. Now, in this situation, you have your top-down goal, which is looking at the road ahead. But then there's also the bottom-up saliency. There's stuff that is attracting is calling for your attention. And then we ask sort of, how does the competition unfold in visual cortex between your top-down goals on the one hand and your bottom-up saliency on the other hand. So to study this, we use a paradigm that has been used a lot and has been developed by Jan Theeuwis a couple of centuries ago by now. No, that's not true, but at least in the 90s. I hope he's not listening. Which is called the additional singleton paradigm. And here, the target people have to find is the odd shape out. So that's, in this case, a diamond, could have been a circle. And then they have to report the orientation of the line as fast as possible. So this is basically the road ahead, right? They're looking for the odd shape out. That's the road ahead, what they have to focus on. But then we also introduce salient distractors, which are the odd colors out. That's basically the flashing billboard, if you will. And then all the rest are these non-singleton distractors that are not particularly salient, but are there as well. So every trial would always start with sort of placeholders, and then suddenly a display like this would appear. And what's important is that people did not know a priori what shape to search for, right? Could be diamond or circle, it's just the odd shape out. And they also don't know what distractor to suppress. So they could not generate any like preparatory templates or initiate proactive suppression. So we're really measuring competition between these top-down goals and bottom-up saliency as it unfolds in real time. Now, what is nice is that since we're using EG, or you could also use MEG here, is that you have the temporal precision of EG and MEG. But since we know exactly what locations we're tagging, we also have like the spatial specificity of methods like MRI or whatever. We know exactly what stimulus we're tagging. So in some trials, the distractor was present. On other trials, the distractor was absent. And what is crucial is that we then tagged either the target or the distractor. And this could be then a salient distractor, or when there was no salient distractor, it would be the corresponding non-salient distractor. Of course, everything is counterbalanced. You can trust me that it is when I don't say so. Now, just as a bit of a sanity check, let's look at behavior, like are these salient distractors really distracting? So we look at the response time to the target when the distractors are present or absent. And what you see is that people are a lot faster when there is no salient distractor and a lot slower when there is a salient distractor. So yes, salient distractors are distracting. That's good. Jan would be happy. Does the tagging work? That's important. So let's look at the spectrograms. So here you can clearly see that at time point 0, where we initiate the sinusoidal luminance modulation at 60 Hertz, you see a clear peak at 60 and only at 60. Typically, you always find those in the back of the brain in like occipital, occipitoparietal electrodes. And then for the 64 Hertz, we also find this clear 64 Hertz modulation after time point 0. Now note that we tagged both of those simultaneously, but they show up in separate analyses because we randomize the phase of the tagging, which allows for better source separation between the two. So in any case, does the tagging work? Yes. Lots of sanity checks today, but today I'm going to show you a lot of graphs like these, where on the y-axis there's coherence. That is basically to what extent does my riff response resemble the luminance modulation of my tagged stimuli. And you don't need to know a lot about this other than just more is better, to keep it simple. So first we're going to compare the response to the target to those of all these sort of uninteresting non-singleton distractors. So here in solid lines, you see the response to the target, and the red indicates the difference. So you can see that already after like, I don't know, 1 or 2 hundredths of milliseconds, you have a stronger response to the target than to all of these uninteresting non-singleton distractors. It's good. Target is attended, you could say. Then we can also compare like the salient distractor to these non-salient distractors to see whether the distractor is attended as well. And also within this very early timing window, you have a stronger response to the singleton distractor than the non-singleton distractors. So it's good. Distractors are attended as well. Now it becomes more interesting because what is the fate of the target? Like how strong is the response to the target depending on whether or not there is a salient distractor. And actually, very early on already, there's a difference between those two. So the presence of a salient distractor downregulates the very early response to a target. So it weakens target processing. And now what happens if we compare the response to a target versus a singleton distractor? Now note that they are both singletons, right? One is a shape singleton and the other is a color singleton, but one is relevant and the other is not to participants. So at very early time windows, it seems that attention is initially equally distributed between the targets and the salient singleton. Maybe we expected slightly stronger response to the salient singleton, but that's what it is. As a side note, for the salient singleton, for the distractor, this peaks a bit sooner than for the target. So there seems to be a difference there, but not so much in the magnitude. But anyways, they're both singletons, so I'm happy with either. And then at later time windows, what you can see is that the target overcomes the salient distractor and then wins the competition, right? So over time, the target is attended more. And what I liked about this study in particular is that if you look at the specific difference between those, so really the measure of competition between target and the salient distractor, this difference score actually predicts subsequent response time. So we're not measuring some epiphenomenon. It predicts how fast people are going to be able to respond to the target.
Surya, can I just ask a clarification question?
Yes, of course. Yes.
So one is mine and one is somebody in the chat. So I'll first do Joaf, who asks, is the detection of rapid stimulus modulation confined to early visual areas, V1, or does it extend to higher order processing stages?
Yeah, so what we typically do is we first figure out which electrodes show the strongest rift modulation, and those happen to be posterior electrodes. However, if you look at the entire scalp topography, I have a couple of results where I do show those actually. Actually, this sort of, you find it throughout the cortex, but that is mostly because those electrodes are highly correlated, not necessarily because this is the origin of the signal. Because some MEG studies with source localization have shown that this rift response is mostly confined to V1, V2. And people like Matthias Müller will tell us that after a couple of synapses, these 60 hertz modulations die out.
Okay.
So the rift response itself should be more or less confined to early visual areas.
So it's really a measure of kind of feed-forward processing strength.
Yes, exactly.
Then another small technical question is, in these plots over here, you have above baseline coherence before the onset of the stimulus. What does that mean?
Yeah, so we never sort of baseline correct our coherence. It's just because whenever you feed any noise into a system, just by chance, you will have some overlap between let's say, a random signal and your sine waves that you're interested in.
Thank you.
It's just that it's not big. We could baseline correct it, but you clearly see the onset, so that's what it is. Thanks. Let's see. Yeah. So a second ago, I showed you that sort of at first a distractor, salient distractor shows a stronger initial response than a non-singleton distractor. And what's interesting here is that if you look at later time windows, you actually find active suppression of the salient distractor. So this was something quite nice that has been found in some studies, but not in all. So you really find this sort of complete time course where first attention is allocated towards the salient distractor, but then later the salient distractor is actually suppressed to below the level of the rest of the screen, if you will. So in this project, there's still a QR code in there. It doesn't work anymore for all these studies. You can just look at my CapLab website. I'll share the link later. But here in the studies, we show how sort of these rapid attentional dynamics unfold when top-down goals compete with bottom-up salient events. And it's really only with Rift that you can sort of track those simultaneously and know exactly from which stimulus these responses come from. And specifically, we see that people first attend the distractor, then suppress the distractor, and this leads them to attend the target. So in the next project, this is one that was run by Lasse, who is also a PhD student in my lab. He looked at perceptual competition over time, right? Stuff is not always relevant. Sometimes we know when things become relevant. So to give you a bit of an idea, when you're at the bus stop and you're looking down the road, whether the bus is arriving, and you want to know whether this thing at the horizon is really a car or your bus, this is only relevant when your bus is supposed to arrive, right? So when your bus is supposed to arrive 10 minutes from now, this is not very relevant visual input, but when you know that your bus might arrive at any moment in time, then it makes sense to compare this template that you have of a bus to the visual input. So then the question is, do we sort of crank up visual processing in anticipation of relevant visual events? And the way that we studied this is with a task. It looked a bit like this. Not all participants had mustaches, by the way. First, we provide the identity of the target. So basically, well, this is the bus that you have to look for. This was a grating that could be oriented in any bunch of ways. And then we provide them with a cue. That's basically the timetable telling you the bus will arrive early or late. So it will arrive at time point 1, or the bus will arrive at time point 2. And if it arrives at time point 2, you know exactly when the relevant visual events will arrive. The event that arises before that time is irrelevant to the participant. Right? So then in this case, it's the second grating that is subsequently presented that is the relevant timing. That's when the bus can arrive. So here participants have to say, hey, was this my bus or not? Was this the same grating or was this not the same grating? And then they provide a response. In this case, it's different, right? These were different orientations, so they say different. Now, as a sanity check, we first look whether we can distinguish between memory matching, so same visual events, and memory mismatching, so different visual events. And whether it matters whether these events are then at the relevant moment in time or at the irrelevant moment in time. Because you would say that whether something is your bus or not would only matter when this is the time at which your bus should arrive. So we're going to look at the time relative to these probes, so these orientations. And here what we do is we decode between whether they match what you're looking for or they mismatch what you're looking for. And what we see is that at the relevant timing and the irrelevant timing, in both cases, we can decode whether this is your stimulus or not, whether it matches or mismatches. But this decoding is a lot better when this is at the relevant timing. So exactly how you would expect. So people are doing the task, and this is also reflected in sort of normal multivariate decoding. So we find better multivariate distinction between matching and mismatching stimuli. Now, the interesting thing, what we can do with Rift, but not with your traditional decoding, is to see what happens in anticipation of these events. What happens before a probe is presented, depending on whether it's relevant or not. Do we crank up visual processing in anticipation of a relevant visual event? Now, of course, this task was not only presented like this on the screen, but we added Rift tagging throughout. So there was a sinusoidal luminance modulation of this central location throughout the entire experiment. And then we can see what happened in our task. So in the x-axis, we have our events. So first there's the memory item, and then you have your cue, first probe, second probe. And again, on the y-axis, we have our coherence measure. So tagging goes on, and then, of course, you find coherence, as you should. But then we have a different expectation depending on whether the cue was a 1, so you expect a relevant event immediately in dark blue, or whether the cue was a 2, so you don't expect a relevant event immediately in light blue, but later on. And then what you see is that they start to separate. And then if you plot the difference, then indeed this is a significant difference between those two. Now, in the second time window, you should now see a reversal, right? Because now the light blue is where the subsequent stimulus would become relevant, whereas for the dark blue situation, when a 1 was cued, it was no longer relevant. And then indeed we find this reversal. So it's nice to see this pattern flip. And it also all happens in these posterior electrodes where we typically find these Rift results. So to find sort of a general answer to our question, we can sort of combine these two. So we just pulled these two periods together, and we see indeed like a very substantial proactive increase in visual processing in anticipation of relevant visual events. Okay, so this is something that Rift could reveal, but you could not do with decoding. So there's a relevant event that people anticipate, they crank up visual processing, and then subsequently this increases their perceptual discrimination between the matching and the mismatching stimulus.
Surya, can I ask a quick question about that?
Yeah, of course.
So is that generalized across the whole visual field? I'm wondering how spatially localized this kind of ramp up pre-stimulus is.
Yeah, so in this particular case, we only did it at the center because that's where sort of the risk response is stronger. So that's where I wanted to start. I don't see any reason why that would differ because all like attentional modulations work similarly across the visual field, just with a little lower SNR. But that's a good question. But what I like about this is when it's sort of peripheral, the cranking up could also be just due to sort of spatial allocation of attention moving towards that location. Whereas here, anything that's happening is already at the fovea, so people don't move their attention. So that's why we can really isolate sort of, well, let's call it sort of the cranking up of visual processing. And otherwise, it could not be disentangled. But there's no reason to believe that it would not work at the periphery,
No,
unless
I
you
guess
have
my
one.
question, it wasn't about whether it would work at the periphery. I guess how spatially diffuse do you think this ramping up is? Is it just kind of a gain control across the whole of vision, for instance, or is it actually focusing in the
Yeah,
in
I mean,
the
I could just give you an educated guess, and I think it would be quite spatially segregated.
yeah,
But the truth is, we haven't run that yet. But as long as you spatially segregate it, then you get the problem of sort of spatial attention also playing a part in it. So we probably would have to come up with a smart trick to dissociate between these two.
right. Okay, thank you.
But that's a good point indeed. Yeah. Um, Okay, so the next project is led by Kabir, which is also a PhD student, a brilliant PhD student actually, and he's the one who built the Rift Lab. So he spent like the first 1.5 years basically doing an engineering job before he could finally start collecting some data. And he looked at perceptual competition or attentional competition, if you will, in perception and memory. Now, to give you a bit of an idea, imagine that I would ask you, what is the exact color of your phone case, right? So if you try and think about the color of your phone case, it's very likely that you actually shift your spatial attention to the left or right, depending on which pocket your phone is in. And this is although the location of your phone is completely irrelevant to its color. This is just something that has been found, right? And it's believed that human observers use a spatial reference frame maybe to organize their memory content. Now, in the case of sort of external attention, right? So like here, when your phone is actually there, if I ask you about the color of your phone, there it would actually make sense to allocate your attention to the side where the phone is, because then there's actual visual information to boost through attention, right? So there it actually makes sense. And this situation is the traditional like external attention, but the situation of the phone in the pocket is what is called internal attention. And they're believed to rely on very similar mechanisms because in both cases you direct your attention to one side as opposed to the other. But in one situation, this is actually useful for sampling information because there's still information there to attend, whereas in the other situation, it is not useful for sampling information. So we asked whether attending a location always boosts visual processing at that location, or only when there's visual input to be boosted there. In other words, does Rift track internal attention to the phone in your pocket or only external attention to the phone on your table? Now, the way we studied this was in a simple task. People had to memorize two colored gratings with different orientations. This should appear later on. And then they had a cue, for instance, a blue dot, and that would tell them that they would have to memorize the blue item for later recall. So they already memorized both, but only the blue one would be relevant for subsequent recall. So the hypothesis is there, if people use a spatial reference frame, once the blue cue is presented, people might move their spatial attention towards the former location of the blue stimulus. Even though blue is not relevant at all, this location is not relevant at all, they just need to report the orientation of the blue stimulus. But the hypothesis is that people do this, that they, when seeing the blue cue, they move their attention to the previous location of the blue stimulus. And then they report its orientation. So this is to test sort of the internal attention situation. And then we have a matching task in which people first have a cue. So in this case, they know that they're going to have to report the orientation of the blue stimulus. Then the two stimuli appear. So at that location, at that moment in time, they know that they have to attend, in this case, the right stimulus, the blue one, and then they report it. All right, so here at the top location, it's not very useful to direct your attention to the right for sampling information, but in the bottom situation, it is useful because there's actually information that you need for reporting. So how do we know that there are shifts of spatial attention to begin with? So we have a couple of markers of spatial attention, and the first one is alpha lateralization. So these are not induced oscillations, these are just spontaneous oscillations. So we take the alpha power and then we subtract trials with item queued on the right, with item queued on the left, and then we find this very nice lateralization. So this shows that in both the pre-queue task and the retro-queue task, the queue caused them to shift, well, at least caused an alpha lateralization. Let's keep it there, right? So this is an indication that there is some shift of attention happening, although of the same magnitude, although it was useful in one task and not in the other. That's good. Then we can also look at gaze bias, something that Freek van Ede has been using a lot. And just sort of to cut long story short here, we find very similar gaze biases in both tasks. Right, so this is another marker of attention. People appear to really shift their attention to the queued side in both cases, even though in one situation it's not useful for the task at hand. Well, we have a couple of other markers, but I think that that's enough for now. So yes, there is a clear shift of attention. But now what we ask is there also like a change in a rift response at the attended location in both cases? Right, so covert attention should shift to the blue item in both cases. So that means that the rift response should be higher at that location. But we're not sure whether that will also happen in the case of internal attention when there's no information to be boosted there. So here again, we can just retrieve nicely the two frequencies that we used. They're all very nicely located in parietal electrodes. And now the critical thing is that in the pre-queue task where there was an actual stimulus to boost, we find this very clear rift response. So there's a stronger rift response at the attended location than the unattended location. But then in the retro-queue task, nothing is happening. So when we saw this, we were like, okay, apparently early sensory processing is selectively enhanced for external attention when there's stuff to boost, but not during internal attention. However, there's also a difference between these two conditions, is that here we're tagging an empty location and here we're tagging a stimulus, right? So we're not sure, maybe we're just not able to tag these empty locations. So we added a very similar task, which was just with a spatial cue, which is just attend right or attend left. So here we should find a change in rift response between the attended and the unattended side, but there's no stimulus to tag. We're just tagging gray background. And here we again find all the markers of attention, and more importantly, we find this very clear and very sustained rift response again. So we do find, based on these sort of standard markers of attention, that memory uses this spatial layout, even if it's not useful, but it's not reflected in early sensory processing, right? We only boost our sensory processing when this is useful to sample new information. So this is not the same mechanism as the one that is used for external attention. So to summarize, RIFT provides a measure of early sensory processing, but it's not modulated by internal shifts of attention. We find this location-specific bias in early visual processing. We find this increase in what some people have called cortical excitability, if you have ever talked to Ole Jensen, who is relatively close by, I think. And we find this even in the absence of a perceptible stimulus, but only when this is useful for perceptual encoding. Now, the last situation that I want to discuss is about perceptual competition for stimuli that actually occur in the same region of visual space. This is perceptual competition during binocular rivalry. I think this is a phenomenon that most of you are familiar with, but just to give you a quick heads up, this is a study done by research assistant Koert Stribos. Binocular rivalry happens when you present two different stimuli on a screen, but to the corresponding locations of the two eyes. So in this case, we presented, for instance, a blue grating and a red grating to two different eyes, but then we tagged the two stimuli at different frequencies. So one was tagged at 69 hertz, whereas the other one was tagged at 60 hertz. And then, of course, everything was balanced and so on. But then using a mirror stereoscope, we ensured that both would overlap and appear in the same region of the visual field. And because of that, binocular rivalry is evoked, and then participants see not a combination of both gratings, but they see first one grating and then the other, and then one and then the other, and these alternate over time. And here we ask whether we could actually track this type of binocular competition in early visual cortex using Rift. So participants had to report whenever they saw the, I think, left tilted or right tilted stimulus. I think the response was based on the orientation, if I remember correctly. And they did this for quite a number of relatively long trials. And there have been a couple of studies using a flicker frequency and binocular rivalry, but the big problem there is that we know that sort of intermittent stimulus displays really screws up with rivalry dynamics. So the same logic that I had with covert attention also applies here, is that we have no idea what happens during binocular rivalry when you just have sort of the natural ongoing process occurring. Just for a sanity check, we see that sort of the percept durations follow the gamma distribution as they typically do. And also there was no difference between the 60 and the 69 hertz responses. So the different rift frequencies at least were not affecting the dominance duration. So to analyze the EEG data, what we did here is that we looked at the manual responses where people said dominant, recessive, or basically they said leftwards, rightwards. So we know when each stimulus becomes dominant or becomes recessive, because when one is dominant, by definition, the other is recessive. And now for the key analyses, we normalize time around a switch so that we have an EEG response for a dominant percept at time 0, and for a recessive percept at times -1 and +1, with all time points in between being interpolated linearly. Now remember that these stimuli are both continuously presented, so nothing is changed in terms of visual input. The only thing that fluctuates over time is which percept is dominant, right? So this is really, can we track conscious perception using Rift in early visual cortex? And then what we see is, again, on the y-axis, you have Rift coherence, is that you find this very nice progression where sort of the recessive stimulus evokes a very much weaker response than the same stimulus when it is dominant. And again, we find this mostly in occipitoparietal electrodes. Now, this is normalized time. So maybe for rivalry, in this case, it's interesting to know what happens in sort of actual time, because how long before a report can we actually see that there is a switch in the EEG response? Now, this is the same graph, but then in raw time. So without interpolation, without time stretching, and so on. So of course, the response to the recessive percepts are smeared out because of the variability in response times. But here we can see that by testing where the derivative of the signal differs from 0, that almost 2 seconds before participants reported a shift, the Rift response to the stimulus that is about to become dominant already starts to increase, and then peaking about 300 milliseconds before the report, which is, well, probably just their response time, I would assume. So this is very nice because now we have sort of this first evidence that Rift can track perceptual competition during binocular rivalry. And this is a line that we are pursuing here, because now we can ask a lot of different questions. For instance, can we use this in no response paradigms? So we're currently trying to sort of classify the dominance versus recessive percept based on the EEG alone to see whether we needed those responses to begin with. This is somewhat promising, although a bit noisy, but this is something that we're currently working on. And what I'm also very much sort of wondering is whether binocular rivalry actually takes place, sort of from neurally speaking, when there's just a gray screen presented to both eyes. I have no idea, but that's something that I would find interesting as a rivalry nerd, so to say. Let's see how I am on time. I think there's still some time to cover a couple of forward-looking ideas. So what is the future? So there's a couple of things we're working on now. And one is brain-computer interfaces. So in this case, this was Rift with overt attention. So people could just make an eye movement towards one stimulus or the other. Please note that, of course, on the screen, the flickering was invisible, but for you, there's sort of the refresh rate of my monitor and your monitor stacked on top of it, and you can clearly see the flickering. But this is the little marker is moving just on the basis of the Rift coherence response while people make eye movements. Ideally, of course, eventually people don't have to make eye movement and it's just based on covert attention alone. But this is also something that we're working on. Now, in all the studies that I showed you, we tagged, I think, only 1 or 2 or maybe 3 locations. Now we just a couple of weeks ago, this data came in where we were able to tag 5 different frequencies, but by also varying the latencies, so the phase of these different frequencies, we're now actually able to tag 10 different frequencies. And this is one single participant, one session. So we can already very nicely tag 10 different locations. And I guess the goal eventually, the dream I would say, is to be able to tag the entire visual field, I guess the same way a gradient does its job in an MRI. So that would be good because then you would have sort of a real life tagging of the entire visual field that's sort of increasing visual precision. Another thing that would be nice would, of course, to collaborate with you because where Rift started out a couple of years ago, you needed this very expensive ProPix projector and very expensive MEG. Well, that doesn't make it too easy to sort of just include it in sort of all your day-to-day experiments, right? So the first thing that we changed here is that we found out that this works just as well with EEG. And the second thing is that now we're also using gaming monitors that are at 480 hertz. And these gaming monitors, we also recently published a preprint showing that we can actually track a covert attention on these gaming monitors at 360 hertz and at 480 hertz. So that suddenly makes Rift a lot easier to use in your actual experiments that you already have. You can just include it in your experiments if you have a 360 hertz monitor, which are not that expensive. And if you're interested in sort of controlling for attention or so, just have your stimulus flicker and it doesn't change anything for your participants. So that's quite nice. And it's quite cheap with EEG and a gaming monitor. So that's why I was trying to coin the term ghetto Rift or Grift, but it didn't catch on so far. But maybe it will work now. And in helping you also to do these Rift studies, Kabir, but also together with a bunch of people from Nijmegen, from the Donders Institute, we sort of wrote this collaborative guide to rapid invisible frequency tagging, which you can find as a preprint also on my website. It has tons of things that I'm not going to cover here, but it's quite nice because it explains you how to build your lab, how to build your stimuli. There's also code that you can use and you can see how to analyze the data, what are all the problems that we encountered and the solutions. So if you're interested in doing that, I really advise you to have a look on my website and check that out. So to allow to still have some time for discussion, I want to thank all sort of the collaborators here, in particular Samson Chota, who is the co-PI of this sort of sub-Rift lab within my CAP lab, and Kabir, the PhD student, who is really the driving force between all of the Rift stuff that we've been doing. And this is my CAP lab. Please do note, I do normally not force them to wear a cap, but for this picture I did. And do have a look at my website for all the preprints and papers. You can all find them over there. And having said that, I want to thank you for your attention. I think I'm going to minimize my screen so I can actually look you in the eyes instead of sideways for the discussion.
Awesome. Thank you so much, Surya. Let's give Surya a round of applause. Yay. That was
And
amazing. That was very cool. I have lots of questions, but I will
did
first
I
give the
stop
floor.
sharing already? Oh yeah,
Yeah,
okay.
you did. You did. This is all good. We're seeing everybody now. If you have a question, please use the raise hand function. I will first start off with one question that's in the chat that we have from Rob, who asks, cranking up and ramping up, is the nature of the label localized mean firing rate modulation or temporal offset? Is that clear? Otherwise, Rob, could you maybe clarify?
So I guess the truth is that it's not entirely clear. So until now, there are a couple of labs that are using the term cortical excitability. We use a term sometimes very vaguely, enhanced visual processing. So basically, depending on sort of the people that we're talking to, we want to speak their language and say something that is useful to them. What I know and what I like is to actually stay quite close to what it is we're measuring, and that is just a visual evoked response, right? We sort of show a stimulus, and then you have a response as a consequence of that stimulus. Show many stimuli, we have many responses to that stimuli, and we know for sure that it's a response to that stimulus because of the exact timing of the response. So I would also be able to call it like responsivity or maybe how prone the visual system is to respond to it. But the truth is that we don't know a lot much beyond that. So that's a bit of an unsatisfying answer, maybe.
Well, no, that was very helpful. Thank you. Another way to rephrase it, perhaps, is the coherence can either be generated by literal coherence, better time phasing that results in a stronger signal, without changing spike frequency, mean spike frequency at all in V1 or V2 or even LGN for that matter. And I think there's a presumption in the field to assume that information is carried by mean firing rate, which I think is a wrong presumption. And I'm just wondering your thoughts on, could it just be changing phase lag and phase advance on signals?
So as someone who used to sort of study sustained working memory representations during functional MRI and finding substantial decoding, then moving to EEG and seeing that there's no decoding left, the idea of yes, I thought it was all activity and now it turns out not to be, is something that is a bit painful to me. And also something that I'm not sure I would dare to venture too much into. Now, what I do know is when you're analyzing these responses, you can also break it down into sort of the phase-specific part of the signal and the amplitude-specific part of the signal, which is then phase A-specific. And typically, we find all of our effects in both. Most of them are captured by the amplitude, but they both really contribute to the eventual signal. So that might answer your question in a way that you'd probably know better than I do.
I doubt that very much, but thanks.
Awesome, thank you. Steve.
Yeah, thanks for a great talk. I think it kind of builds on that discussion. So I'm wondering how you think of the potential interaction between these tags and any endogenous rhythms that are going on, alpha, theta, and so on. So I guess the assumption in the analysis is that it's just like you're paying the system and you can read that out, but presumably there are interactions happening as well into any endogenous rhythms, especially in relation to things like attention.
Yeah. So there's two important things there. I think it's a very valid point, by the way. And there, in Ole Jensen's group, what's her name? Catharina Duker has spent a lot of time in trying to see whether you could entrain these neural oscillations through Rift, and that's really not the case. So at least inducing those 60 hertz rhythms doesn't cause any sort of 60 hertz rhythms beyond sort of where you stop the stimulation.
Mm-hmm.
So that's one thing. And also she couldn't find any sort of predictability between sort of amount of 60 hertz before and after the start of you inducing these signals. The second thing is that what we do is we, to separate the signals in most of these studies, we also randomize the phase. And as you saw in some of these plots, is that with randomized phase, the coherence of our particular signal completely vanishes. So the coherence measure is very sensitive in that regard to phase, because that we use to disentangle signals. So any endogenous rhythm at 60 or 64 hertz would not be in line with sort of the stimulus that we present at 60 hertz, and thereby it's canceled out in the analysis given sufficient trials.
Okay, thanks.
Chris.
Yes, I was very interested in the work on binocular rivalry. And I'm interested in the idea that perhaps binocular rivalry can happen in the absence of consciousness, so that your report-free method would be very interesting. Have you any plans to look into that possibility?
Yes, I'm so happy that you say that, because here I was the only one interested in this. But for me, that was indeed where I wanted to move towards. So I know that, for instance, Jan Braskamp has this Nature Neuroscience paper where they presented two different sort of dot stimuli that would rival, and then you would see that a perceptual sort of binocular rivalry would continue, even though participants sort of don't really know whether one stimulus or the other is dominant, because they look so similar. But
Right.
there is still local conflict because the stimuli are different perceptually, right? There's either a dot here or not. And indeed, what I wanted to move towards is indeed to end up with sort of having this situation of a gray screen in both eyes. But one gray screen is actually sort of 60 hertz going from black to white, and the other 64 going to black to white, and then see whether we still find this gamma distribution of rivalry distribution. So indeed, for me, this was the end goal of this project, which means finding a new research assistant, because my colleagues are less interested in it than you are and I am.
Thanks.
Amazing. I have a follow-up question on the binocular rivalry as well. I thought that was super interesting. Do you feel like your results go against the idea that binocular rivalry is feedback initiated, which is stuff that's been found by Fiet Wildhammer, for example, where they see the inferior frontal gyrus causing these shifts? How do you see that?
No, yeah, no, I don't think so. I mean, I think generally, isn't it? I think it's the case that there's in general more feedback projections than feed forward projections anyway. So the fact that you find something in early visual cortex doesn't mean that it's not feedback, especially during binocular rivalry, where you know what the other stimulus is. You have seen it already. So there's all types of predictions and knowledge that can play into that and have been shown to play into that. It might be different, for instance, if we would use something like continuous flash suppression, where the sort of stimulus that is recessive has not been seen yet. So there you could not know what it is, and you could not use your knowledge or predictions to sort of boost it in a particular way. But in the case of rivalry, you know exactly what the other stimulus is. So yeah.
I guess the reason that I'm asking is because it seems like Rift very much represents the feed forward signal, the kind of amplitude strength of the feed forward signal. So I'm thinking, would the idea be that you have a top-down prediction, which then biases the kind of the sensory precision on that specific content, and that then leads to the increase? Is that
it has to, right? Because I mean, even sort of beyond this whole Rift stuff, I guess sort of all the Randolph Blake studies and everyone who copied that same work with flash suppression later on, like me, they really all show that sort of rivalry is manipulated by all types of high level cognitive processes, right? So you have attention, expectations, working memory, semantics. They sort of all bias binocular rivalry, and then you even find sort of these rivalry dynamics in LGN. So I don't think that sort of the semantic meaning of an image would be sort of extracted in LGN. That would be very unlikely. So it's very clear that those are feedback projections. Yes.
Yeah, that makes sense. Rob just asked another question in the chat about Adam Silito's work.
Yeah, let me give you a little prelude on this because
Please
you mentioned
do.
LGN just now. So he basically spent 20 years looking at what layer 6 neurons in V1 and MT were feeding back to LGN. And you can actually record orientation selectivity in LGN. You can record motion, background, subtraction, and you can record it all in the LGN. And layer 6 neurons in V1 are sort of at a position of control in the fact because they drive 40% of all excitatory drive to LGN is from those neurons, and those same layer 6 neurons also drive layer 4 neurons in V1 more forcefully by a factor of about 4 in terms of synapse number than does the LGN itself. So my question is, it seems like your work is right on the cusp of kind of contending with the neuronal mechanics of how this is implemented. Or you're at the interface where we want to be between the psychology, the binding, and how it's done. And I just, if you know anything or read Adam's work or the inactivist tradition, which is also retrograde from inside out rather than from outside in. I'd love to hear your thoughts on
Yeah,
that.
so I think on the one hand, I think I have to disappoint you a bit because I don't know his work, or at least not by name. So it might be that I've seen the work, but not know the name.
University College London. So yes.
Okay. So I think that there's this sort of, I guess, a tradition in neuroscience and in psychology that when we have a new method, we sort of immediately start answering big questions like, oh, we just discovered fMRI, let's answer consciousness. Whereas in physics, they would sort of first measure everything to the core of it and then start asking tiny questions. So here, as I said before, there are still a lot of things that I don't know exactly about how it is measured. So I would also be maybe a bit careful in making very mechanistic interpretations of it. And one reason for that is that although on the one hand, there's people like Matthias Müller who say, well, after a couple of synapses, these 60 hertz oscillations are gone, so it has to happen in V1, V2. MEG source localizations also tell you that. But then there's also some work by Linda Drijvers at the Donders Institute, or actually Max Planck, Donders, well, somewhere in Nijmegen, to the east of me. And they found that if you have an auditory stimulus at, let's say, 68 hertz modulated and a visual stimulus at 60 hertz, you find intermodulation frequencies somewhere in frontal brain regions. Now, this is not possible. These intermodulation frequencies would not be possible if the auditory and the visual responses would not somehow be combined at some point. So either the auditory stimulus propagates all the way back to V1, which I think is unlikely, or the visual stimulus propagates forward to frontal cortex, which is highly unlikely to others. But there is still something very unresolved here, because that you cannot have an intermodulation frequency of two stimuli, two sinusoidal modulations that don't meet. So this is just to say that there are some things here that we don't sufficiently understand yet to make mechanistic claims about neural implementations. Long story short for meh, not sure.
Thank you. Yeah, thank you.
Excellent. Anybody else have any questions before I dive into some of mine?
Oh, that sounds frightening.
No. I just had because I think it's fascinating. The two things that I was most excited about was the one finding where you found that you get these modulations in alpha and anticipation to a memory or kind of for spatial location of a memory, but you don't get this modulation of the rift response. And I thought that was really interesting, and also intuitively, and I wonder, I think maybe we had this discussion before, I would have expected a suppression of the rift response in those locations.
Why suppression?
Because I would expect that when you are attending, internally attending a specific location, you want to inhibit the input at that location to protect your internal representation so it doesn't get overridden by whatever is coming into your eyes.
So I think that would be also a bit that this could be related to what Steve said before. So maybe if people sort of direct their attention to sort of internal representation, so to working memory, they might have an overall reduction of whatever is happening externally, right? So that would show not on the left versus right contrast, but like an overall reduction. But we don't have the conditions to contrast that here. So here, the only thing we can compare is sort of the left versus right, rather than internal versus external attention, which is also an interesting situation. But that would resemble maybe sort of this timing study a bit more.
Yeah, that's interesting. So your expectation is maybe it's just not location specific.
Exactly.
It's like a general tuning up or down.
And so
Yeah.
what I think this, I mean, people have discussed this before, right? It's just easier if you memorize two items, visual items. This is just a lot easier if you sort of juxtapose them in your sort of mental world, if you will, than superpose them. I mean, it's very difficult to do that. You would not do that. Even
You
for
mean
long
sub
term, yeah, so if you have to memorize two great things in your mind, you will not memorize them superposed on top of each
Mm.
other, sort of at fixation, because that would just make life more complicated, and also in terms of neural competition. And I always see this a little bit as the short-term memory equivalent of the memory palace, where people do these long-term memory competitions, but then they sort of create this space because it's just easier to sort of retrieve and position these stimuli over there. It's not the same, but I see this as the short-term equivalent.
Cool. Thanks. Alexander.
Yeah, thank you. I guess I just would like to clarify this moment about memory and shifting. So if I understand correct, do you mean when we shifting from right to left, is it accurate to say that it's tricky spatial temporal reviewing of location? So does it mean that when we work with abstract meaning of something, abstract symbols, we located this the same in a space? Can be located not only visual stimulants, but when we talk about something and people move gaze right or left, so they connected meaning with space too.
Yeah, this is interesting. So I think in case of visual stimuli, right, I would say it's just easier to organize and thereby memorize things in your working memory if you give them a location. And when we as experimenters have already given a location by the means we present them at first, then people might use that location. If you give them non-visual stimuli, I guess one of two things can happen. People might make them visual because we like vision. That's one possibility. But I'm not entirely sure if you give me very abstract concepts, like, I don't know, some very specific type of emotion versus some other type of emotion, whether I would also organize them left and right visually. That's an interesting question. I would tend to say that we like to organize things in a spatial manner, but that would be a guess, and your guess would be just as good as mine in this particular case.
I just would like little comment. I do microphenomenology interview and notice how people connected some abstract meaning and make movement of the body and gaze. And usually when they try to locate it something in memory, they do like right and left, right and left.
Yeah.
So for me, it was question, does it connect or not? Anyway, thank you.
Yeah, so I think it is connected. I think we are very good at spatial processing. We do that all the time, right? So I can imagine that sort of a lot of the other cognitive processes may piggyback on sort of that incredibly large swath of the brain that is dedicated to spatial processing.
Awesome. Another question I had was on your results where you showed this anticipation of the cranking up of the sensory sensitivity, which
Yes,
was
sorry,
specific.
for lack of a better word.
No, I liked it. It makes sense. And that was location specific, right? I think that you showed.
Well, so in this case, we only had one location. It was all in the center.
Oh yeah, it was stimulus specific, I meant,
Yes,
or like
yeah, exactly.
temporally specific.
Yes.
I was still trying to figure out what this means exactly, this like increased rift response. What does that mean? And I was kind of one way of understanding it is that there's an increased signal of your bottom-up stimulus. But in that, if that would be the case, I would also expect that that continues during the stimulus duration, which I don't think is what you found, and also that it predicts the decodability of that stimulus.
Yeah.
Was that the case?
Yeah, that's a very good point. So what we have seen a lot is that whenever we present suddenly a stimulus on the screen, this breaks down the rift response and thereby also differences between the rift responses in particular. And this may be because a transient has like all of the frequencies in it. It's either the equations that are behaving strangely or it's the actual signal that is disrupted. But this is something that we unfortunately often find. It is the case that you typically see that whenever a stimulus matches memory, that you find a stronger ERP to that stimulus. And I think that's also what we found here, even though normally this is located in space. So we do have like a stronger response to the stimulus when it matches the content versus when it doesn't match, and also when it's relevant compared to when it's irrelevant.
But does the riff
But
predict
in-
that response?
But
Predict
in
the
terms of
strength?
correlations, if I remember correctly, I think we tried that, but that the results were inconclusive, as in Uncle Bass told us, meh. I should
But
say
is
Reverend.
yeah. Okay, thanks.
He
But is
said not
that
no, he said not yes, if I remember correctly.
he was very unhelpful.
Yeah, and also I remember that it mattered a bit, like which time window you would choose, and it became a bit murky. So there we were like, yeah, sort of, we have the causal story in terms of time, but we don't have the actual relations between those.
But is this kind of also the way that you're thinking about it, that it's a measure of kind of strength of bottom
Yes,
up
yeah,
processing?
absolutely. So,
Yeah.
of course, if apparently what we're measuring is that sort of early visual cortex, I hope, responds with increasing strength to stimulation, because then when the relevant stimulation arises, it should respond more strongly to it, which it does based on the ERPs to the relevant as compared to irrelevant stimuli. Now, something actually that we did not expect there is that, so indeed, relevant stimuli showed a stronger response than irrelevant stimuli, but if you would compare matching to mismatching stimuli, this was not the case. We actually had a weaker response for mismatching stimuli, which is fully in line with predictive coding, but which is completely against all the other findings that we ever found where we look at sort of a stimulus that matches the content of working memory. And the reason there is that normally there is this spatial difference. So when a stimulus matches working memory, spatial attention moves towards the stimulus that is attended, creating a stronger response, and away from the non-attended one, diminishing the response. So apparently this typical sort of this difference that we found between what we normally see, you have something in memory, this boosts the response of matching stuff. That seems to be counterintuitive from the perspective of predictive coding. Actually, both are true. So predictive coding seems to be right in that sort of the response per se is diminished when something matches what you already hold in mind. But then because of spatial attention, when there's sort of a spatial layout, then the response is practically increased due to spatial attention.
And you wouldn't find that effect if everything's central.
Exactly. So it was just a coincidence that we found this because we presented everything at the center.
We do find it though in our paradigm when we have imagery instead of working memory. You do get this boost for central
Even for
congruent
central,
stimuli.
that's interesting. I always like to
Yeah.
hope that all these things are the same, right? Feature-based attention and working
Me
memory.
too. I think not always, unfortunately.
Yeah,
Maybe.
we have a review paper coming out about search templates in which we actually sort of disentangle the difference between working memory imagery and feature-based attention.
Awesome.
And I think that they should all capitalize on sort of working memory mechanisms because there is some information that you need to hold on to, but the content of them might be quite different because of the different goals between the different tasks that people give you. So that might explain many of those differences.
Yeah, I guess also like with feature-based attention and search templates, you want to amplify whatever is matching that, right? Because that's the whole point. Yeah. Okay, cool. Thanks. Does anybody have any other questions before we thank Surya again and wrap up for today?
Oh, thank you, Rob, for sending me a package, care package.
Okay, awesome. Well, thanks again, Surya. This was a great first Consciousness Club. Thanks all for coming.
Thanks a
And
lot for all your input and for your questions and remarks and attention. Loved it.
Amazing.
Yeah,
I think we
thanks.
have another Consciousness Club coming up soon, but please keep an eye on the website for that.
Yep, we do. Great to see everyone. Take care.
Bye.
Thanks, Surya. That was great.
Thanks for having me.
All right.
Bye-bye.
See you soon.